15. This is where we'll learn about the principles of disease and epidemiology. Okay, I've provided you with an outline because this is a lot of slides and a lot of topics in this, and it's kind of mixed around in the only a little bit from chapter 15. It's mostly the whole chapter 14. Anyways, here's the outline for you. So pathology, infection, what are infectious diseases, what are contagious diseases, how do we know what the cause of the disease is, all this is what we're going to start with. So an infection means you have a colonization by a pathogen somewhere on or in the body, okay? It's usually a specific area of the body, but we'll see there are some exceptions to that. So a disease is a generic term when the body is not functioning normally, and that could be all sorts of causes, right? So you need to specify infectious disease when you're talking about a disease caused by an infection. This can be also a little complicated because a person can actually have an infection and not have any signs or symptoms of disease. So we'll talk about that later. Etiology is just the specific cause of the disease. So determining, is it a bacteria? If so, what kind? Is it a virus? What kind? So on and so forth. Pathology, on the other hand, is the study of the disease itself. So how did the pathogen get access to the body? What were the steps in establishing the infection? So how did it colonize? And then what are the host pathogen interactions? And what are the specific symptoms of this disease? Why? And usually these host pathogen interactions explain why you have the symptoms that you have. In order to determine the specific causative agent of an infectious disease, we used to use Koch's postulates, which we learned about, recall from chapter 1. But these are really strict rules of isolating. So you isolate the organism from the diseased animal, you grow it into a pure culture, you then take that pure culture, inject it into a healthy animal, and that animal must then come down with the same disease as the first animal, and then you must be able to extract that microorganism into pure culture and determine that it is actually the same microorganism that caused the first disease. And this is really great in that you know without a doubt the cause of the disease, but there are limitations. Not all pathogens can be cultured in the lab. That's the first and biggest drawback, right? There are plenty of microbes that we're not able to work with as far as growing in culture in the lab. There are also some pathogens that cause more than one type of disease condition depending on the location or tissue that's infected. And then of course you have variation in host. Not every single host is going to have all of the identical symptoms. So each model organism also is going to have different outcomes. Some organisms you cannot even infect or cause disease. So this brings up another really important concept that we introduced in chapter 1, and that is that pathogens are host specific. So that's another big limitation, okay? So some pathogens cause disease only in humans because they're host specific. Some pathogens cannot be cultured in the lab, And then you have, what about the different locations of the body causing different diseases, but same pathogen. And then another one that's not on here is that sometimes normal microbiota cause disease. And we're going to talk about what normal microbiota are in a little bit. But, Koch's postulates say that healthy individuals, you must not find the pathogen in healthy individuals. So that would be another limitation. Okay, so how do we classify infectious disease? What's communicable? It is a lot of terminology, all based on concepts though. Okay, what's a sign? What's a symptom? Duration of disease, location of infection, okay, and then this idea of virulence. So, communicable disease means that you can spread it from person to person or from host to host. What is this word host? I've been using probably since day one, and I may not have defined it. Well, as you probably guessed, a host is the organism that gets infected by the pathogen, right? So when we're talking about human diseases, humans are the host, right? You and I. If we're talking about some other type of disease like avian flu, then the bird is the host, right? Things like that. Okay. So you always have to define who the host or hosts are. But in this class, most of the time we're talking about human pathogens and human disease. So usually the host is the human. Some pathogens have complex life cycles, and you'll see that there's actually what we call primary host, secondary host, intermittent host, even accidental host, right? So get used to this word. Okay, so moving on. Disease that can be spread from one host to another, from person to person. Example of that would be HIV. HIV is not considered easily passed from person to person because it's not airborne. It's through direct contact with bodily fluids, such as during sex or IV, intravenous drug use, for example. Contagious disease, on the other hand, is a disease that's easily spread from host to host. These are typically airborne, right? Things like influenza or, COVID. Non-infectious disease means that it's non-infectious. It's not a pathogen that's causing it. It could be something that's inherited. It could be something you're born with, right? Congenital. It could be degenerative. Something that develops typically slowly over time and continues to get worse, like MS, multiple sclerosis, degenerative. It could be a nutritional deficiency, which is probably, if you had to have any type of disease, you would probably want a nutritional deficiency because then you could correct for that. Of course, not if you were in an area of the world where you don't have access to enough food. You could have an endocrine disease. This is a hormonal issue. Could be an issue with one of the many glands in the body, either producing too much or not enough of a hormone that you need to regulate functions in the body. It could be neoplastic. You've probably heard of neoplasia, the uncontrolled abnormal growth of cells or tissues in the body to lead to a tumor or a neoplasm growth. It can be benign, but it could also be malignant. And then, of course, you have idiopathic disease, where a sudden, spontaneous onset of a disease with an unknown cause that is possible as well. Okay. We don't really talk about non-infectious diseases in this class. I don't know if we will talk about them at all or just very rarely. I can't recall off the top of my head. But our class obviously is focused on infectious disease. We do have examples of microbes that are non-communicable, which means they're not transmitted from host to host, but they are still caused by a microorganism. You could get it from the environment. Something like Bacillus anthracis. Oops, that should be in italics. Shame on me. Okay. So you could get it from inhaling spores like Bacillus anthracis. You could get it from ingestion of spores, such as Clostridium tetani causes tetanus. Or for tetanus, usually is from the spores getting into a wound, right? Like the classic example of stepping on a rusty nail. It's not the rust that causes tetanus, it's just that if something's been outside long enough to be rusty, then it's probably been outside long enough to become covered in spores of Clostridium tetani. And then you could ingest Clostridium botulinum, which causes botulism. And you may have heard of Clostridium botulinum from the Botox treatment, where you take the one of the toxins from this bacteria. You put a very, very, very small amount in just very superficially underneath the skin to relax it. This could be cosmetic to treat wrinkles, but we've also developed some medical uses for this, such as people who suffer from chronic headache. Sometimes neck and shoulders need this treatment to get them to stop being chronically tense and causing these headaches. And I think there's other uses for this therapeutically as well. You could look into that if you're interested. So non-communicable, not passed from person to person, but still caused by a microorganism. Okay, so there are symptoms where you feel it, right? Like aches and pains, but we're not really able to measure that. It's subjective. Whereas like a sign would be something a physician or anybody could actually observe and this result of disease. This could be something like a rash, where you look at it, you observe the pattern of the rash, or it could be something like a fever where you actually measure the temperature. So it's really important to distinguish between symptoms and signs, but together, symptoms and signs can really help a physician to diagnose a patient and help get them feeling better. Duration of disease: a sudden, rapid onset that lasts only a few weeks is considered acute. This could be your cold and flu or gastroenteritis. But if it develops slowly or lingers for a long time, then it's chronic. Examples of chronic diseases would be tuberculosis or HIV. Something in between is subacute, right? It's not really chronic, but it's lasting longer than a normal acute infection. And then you have another category called latent, and this is where you have periods of no symptoms, and then you can have periods of symptoms. So the classic example would be cold sores, right? A person has the virus, usually herpes simplex virus 1, and they have that for their whole life, and periodically throughout life they will have an outbreak of cold sores on their lips. They're technically contagious all of the time, but infectivity, or how contagious they are, is very low when there's no cold sores present, and super high when there is a cold sore present. Some people, cold sores are brought on by exposure to sunlight. Others, stress, or if they're ill with something like a cold or even gastroenteritis, like anything that has weakened their immune system and allowed this outbreak. We do have treatments to make the cold sores not as severe and heal more quickly, but we do not have a cure for the herpes virus. Genital herpes is usually caused by herpes simplex virus 2. However, both of these viruses can technically infect both regions, so you can't say definitively that a cold sore is only type 1 and herpes is only type 2. They are interchangeable. Another reason why you should practice safe sex and everything that goes along with safe sex, like knowing that your partner doesn't have any of these diseases. Shingles is caused by the varicella virus. This is the chickenpox virus. And sometimes when someone has the chickenpox, the virus actually migrates into the central nervous system and then becomes dormant for a very long time. And then usually when you're older, like 50s, maybe even 60s, once you have some reason that in addition to being older, you maybe experience a lot of stress, or for whatever reason your immune system is a little weak, then you can have a shingles outbreak, which usually the symptoms are a rash on the back, and it is very, very painful, and you can be very tired. If you want to learn more about shingles, here's a link for you. We do have a vaccine. We have both we have the varicella virus vaccine to prevent chickenpox in children. So children who have this vaccine never have to have an outbreak of chickenpox, and therefore would not be at risk of developing shingles when they're older. Anybody like me who actually had chickenpox as a kid, because the vaccine didn't exist yet, will be advised to get the shingles vaccine. And I can't recall at what age they start, maybe around age 50, they start giving you the shingles vaccine, because like we don't know who has the dormant virus or not, but if we just give everyone the shingles vaccine, then they don't have to develop shingles. Okay. Local infection means the pathogen is in a small specific area of the body. So like a respiratory, usually it's either upper respiratory, nasal, sinus, right? Or lower respiratory, lungs. Okay. It could be a bladder infection limited to the bladder, right? Things like this. Systemic infection, on the other hand, means that it's throughout the entire body, and this is much less common. This would mean typically this means it's in the blood, and that's really dangerous. A focal infection would be an infection that started out as a local infection and has since become systemic. And that is how most systemic infections come about. Rarely do you have a systemic infection that immediately went to the blood, but it does exist. Like, for example, a mosquito-borne disease that just injects it straight into the bloodstream, for example. So we have more terminology around these infections. So sepsis, a toxic inflammatory condition arising from the spread of microbes, especially bacteria and their toxins. Remember, we talked about septic shock caused by the LPS of gram-negative bacteria. Bacteremia specifically means there's bacteria in the blood. It's never good. There should never be bacteria in the blood. So this it would indicate a systemic serious infection. It could be due to an excessive amount of bacteria from a local infection that have then gained access to the blood. Some pathogens actually will travel to the blood. It didn't necessarily cause a huge, infection in one site. Toxemia means there's toxins in the blood, and so this goes hand in hand with like sepsis, for example, where there's LPS in the blood. But it could be toxemia is more broad. It could be any toxin from any type of microbe. Okay, viremia, viruses in the blood, and septicemia means not only do you have bacteremia with bacteria in the blood, but septicemia means they're actually growing in the blood. There are only a few types of bacteria that are capable of doing this, and this is an actual blood infection. We'll see that in our overview of bacteria starting actually this week, and that will last a few lectures long. So something to look forward to. Exciting stuff. Okay, the extent of host involvement continued. Primary infection is the original first infection. It's typically an acute infection that causes the initial illness, such as like coming down with a cold. A secondary infection would be an opportunistic infection that you really only get because you're already weak from your primary infection. So an example of that could be an ear infection, where you have a cold, you have congestion, you have all sorts of now predisposing factors to allow something like an ear infection to happen. Subclinical, on the other hand, means you don't have any signs or symptoms of disease, and you're probably familiar with this concept now because of COVID-19 infections, where we have such a range of people who are severely ill, hospitalized, dying, to people with mild symptoms, to people with no symptoms at all. So if you have no symptoms, you don't even know you are sick, it's subclinical. This is really dangerous because you don't know you're sick, you're not necessarily taking any precautions, and you could be spreading it to all sorts of people who might not be as lucky as you. They might develop the severe form and even die. We first learned about this with Typhoid Mary. I told you about that docudrama about Typhoid Mary. Hopefully you watched that. It will really help you to understand this idea. It's also just really well made. But she was a cook, and she didn't know that she had typhoid fever. She didn't feel sick, but she was still shedding this bacteria, and people who ate her food, some of them would be fine, and some of them would get typhoid fever, and then some of those that got typhoid fever even died. People caught on to this. Public health experts told her, like, you are causing people to die. You need to stop cooking. You need to change careers, do something different where you're not putting people at risk. She refused to believe that could be the case and continued to get different jobs as a cook. So very interesting, kind of scary, kind of sad, but very interesting. So yeah, let me know if you watch that documentary. Next, virulence, another term that you may have become familiar with because of this pandemic. So virulence means the degree to which a pathogen can cause disease. And so we've seen that there's this new strain of COVID, which, although it is more contagious, it seems to be less virulent, causing less severe disease. When we talk about different characteristics of pathogens, we will typically see traits that increase virulence, like having a capsule, right? Having the ability to swarm, and we'll see more and more virulence factors that increase virulence as this class goes on. Virulence of a pathogen can be quantified using controlled experiments and laboratory animals. And so we have these two terms. One's called the infectious dose. ID50 means the amount of microbes that are required to cause disease in half of the test population. And the LD50 is the lethal dose, the amount of pathogens required to kill half of all of the inoculated test animals. And with that, here is a figure. It shows the number of pathogenic agents. Could be bacterial cells or viruses, depending on which disease you're investigating. And then here's the percent mortality in the experimental group. And so as you increase in the amount of pathogens that you inoculate with, you increase the amount of deaths up to half of the population, and then you know what the LD50 is. So a graph like this is used to determine the LD50 by plotting pathogen concentration against percent of infective test animals that have died. In this example, the LD50 is 10 to the fourth power of pathogenic particles. And then you can have a similar graph and do that for the infectious dose, but I didn't include that in this figure. This is from chapter 15. It might not be section 5 anymore, just due to different versions of the textbook. But you could skim through 15 and find where they talk about LD50 and ID50. All right, so now we're going to talk about normal microbiota. These are organisms that live on us or in us and typically are beneficial. So in order to talk about these concepts, we need to identify some terms. So symbiosis means two organisms living together closely, intimately. Okay, symbiotic does not tell you the type of relationship. It just means together. So then we have these additional terms to define what type of symbiotic relationship. So in commensalism, the organism benefits. One organism benefits and the other is completely unaffected. We have some commensal organisms, but I would say most, if not all, of our microbiota are actually in a mutualistic symbiotic relationship where both organisms are benefiting, right? We're providing a warm environment and typically nutrients, and then in return, these microbiota are protecting us from pathogenic microbes. Some of our gut microbiota also provide us with additional benefits. We're rapidly learning more and more about these benefits, but one well-known example would be like the production of vitamins that we actually don't produce ourselves, but we get from our microbiota. Parasitism, on the other hand, a parasite is going to benefit at the expense of the host, and so that's, pathogens are parasites. Usually we don't use the word parasite when we're talking about bacteria. We reserve that for larger organisms like the helminthic worms. But technically, a parasite's anything that's benefiting at the expense of the other organism in the symbiotic relationship. Some normal microbiota become opportunistic pathogens. They will overgrow if they're allowed to and cause disease, or if they get access to another area of the body, they can cause disease there. So a classic example would be a UTI, a urinary tract infection. So you have normal gut microbiota such as E. coli, And if they get access, they're constantly actually going into the urethra, which is why regular urination is important, to clear that out. But if they are able to colonize there long enough, they might be able to work their way up into the bladder and then cause a bladder infection. They could even work their way up the ureters and get into the kidney and cause a kidney infection, which can be life-threatening. So there you go. There's an example. Transient microbiota are temporary. Could be anywhere from days, weeks, or months. Still not causing disease. Normal microbiota we consider to be permanent residents. So microbes that colonize the host at least semi-permanently and also are not causing disease. But again, any of the microbiota could be potential opportunistic pathogens. So, as I mentioned already, we have a mutualistic symbiotic relationship with most, if not all, of our microbiota, playing important roles like protection against pathogens. They're taking up space and resources, which makes it harder for pathogens to be able to colonize. We have the production of vitamins and other molecules, and they can even aid in digestion. So now we're learning that there's actually a really interesting relationship with our gut and our immune system. And so we believe that a healthy gut microbiota from an early age can actually greatly reduce the likelihood of developing autoimmune disorders, even allergies. So really cool, exciting stuff, the field of gut microbiome. So you are actually only 1/3 human, and you are about 2/3 bacteria between all the bacteria that live in your gut, which is the vast majority of our microbiome. But also we have bacteria that colonize our skin, our nasal passages, right? Our mouths. So I already brought up this idea of competition, that microbial antagonism is a competition between microbes fighting for space, resources, right? And so this idea that normal microbiota are protecting us against pathogens because they're already occupying that space, and they might also actively fight the pathogens as well. Probiotics is this idea of live microbes that are applied or ingested into the body with the intention of a beneficial effect. So a lot of people take probiotics to encourage healthy gut microbiome, to reduce diarrhea, gas, constipation, maybe even to reduce allergies. Like, there's all sorts of people are taking probiotics for all sorts of reasons now. Acne is another example. These are not prescribed, that they're not pharmaceutical. We don't have, hard evidence, but we do tend to see a lot of people actually getting beneficial effects. But it is kind of it's at your own, it's kind of an experiment. But I do think that there, especially with the gut, there's got to be benefit from it. But I'm not sure. I wouldn't go as far as to say that we've figured it out how to do that exactly. But anyways, it's a fun idea. Okay. Prebiotics would be things to feed the microbiota. Okay, so there are areas of your body that you will find microbiota, and then there's areas that you should never find microbes, right? So your blood should never have microbes. Your lower respiratory, your lungs shouldn't have microbes, and your vital organs, of course, shouldn't. So Everything else, everything that's coming in contact with the skin, with the air, such as your skin, your eyes, your mouth, your nose and throat, right, the upper respiratory system, your intestines, and the urethra, right, of course, are all areas constantly in contact with the environment and will be colonized by bacteria. This just goes into a little bit more detail of what I've been saying, and it gives you some specific examples of organisms that are commonly found on the skin, in the eyes, in the nose and throat. It'd be good for you to memorize a couple examples. It'll come up later. When we do our survey of bacteria, you're going to have to memorize all sorts of bacterial names and diseases as well. Most of the microbes on your skin are not going to become problematic, but if you get a cut, then they can they can be opportunistic pathogens. Sometimes they get into hair follicles and cause infection that way. Skin is really good at preventing infection by staying really dry, constantly shedding, and having a low pH, as we talked about a little bit. Actually, I might be thinking of last semester. Yeah, you and I probably have not talked about the function of skin yet. So there you go. Low pH, staying very dry, constantly shedding. Skin is a very great barrier. But we also do get little wounds all the time, and that makes you then susceptible to possibly becoming infected, either with your own microbiota or with something from the environment. Okay? So patterns of disease. I'm going to go ahead and make this a two-part lecture. So let's go ahead and stop here, and I'll see you in the next video.